Good morning students. Today we will continue with the types of fermenters what we have started in the previous class or yesterday's class. We will continue with the same topic, that is types of fermentations. And I will again repeat what is fermentation. Fermentation is the anaerobic breakdown of complex organic molecules into or complex organic chemicals by microorganisms is called fermentation. It may be anaerobic or it may be aerobic. Nowadays, for the production of certain vaccines and drugs or medicines, we will be using a low amount of oxygen. That is the reason we are calling it as aerobic fermentation also. Now, these fermentations, based on the production of the product, they are given the names. That is, if ethanol is produced, it is known as ethanol fermentation. If methane is produced, it is known as methane fermentation. If lactic acid is produced, it is known as lactic acid fermentation. Now, see here, that means beer fermentation, wine fermentation, like that. Now, we have seen the two types of fermentations, that is solid state fermentation, or you can call it as shallow fermenters or shallow fermentations. They are useful in the growth of your mycelial microorganisms, that is mycelial fungi, that is Aspergillus, Penicillium, etc. Now coming to the submerged fermenters, and these submerged fermenters, they consist of your stirrers. It is a mechanical one. The solid state fermenter is non-mechanical and the submerged fermenter is mechanical. It consists of stirrers or turbines or impellers or blades which will be stirring the medium uniformly so that each and every microorganism will get all the nutrients. So nutrients and the medium and the microorganisms are uniformly mixed with the help of your turbines. So this submerged fermenter is of three types: batch culture, batch submerged fermentation, fed batch submerged fermentation, continuous submerged fermentation. Batch fermentation already yesterday we have seen with a graph I have shown you, that is growth curve. Now coming to the fed batch fermentation. See here, today's topic is fed batch fermentation. In this fed batch fermentation, see here, this is your fermenter, and in this fermenter what you will be doing is, the turbines are there. In this you will be pouring your culture medium. In the culture medium you will be releasing your bacterial cells or microbial cells, and you will be, this is the inlet, there won't be any outlet. Now you will be pouring the medium. Once medium is poured, the microorganisms are uniformly mixed with the medium, so that the medium and the microorganisms are uniformly mixed. So the cells will enter into lag phase, they enter into log phase, division phase, and the cell number increases. Once the cell number increases, the medium gets exhausted. అంటే ఏంటి, సెల్ నెంబర్ పెరిగేసరికి మీడియం మొత్తం ఎగ్జాస్ట్ అయిపోయింది. వెన్ ఇట్ గెట్స్ ఎగ్జాస్టెడ్, immediately you will be adding the nutrients. Again you will be adding the culture medium. No microorganisms, only once microorganisms are added, and you will be adding the nutrients, media, chemicals. Medium is again sterile medium is added. Again the cells, all the single cells, these small cells, they will start feeding on the nutrients, chemicals that are released into the fermenter, and they increase in size and they start division. Once they start division, the number of cells increases, and as the number of cells increases, the medium gets exhausted because all of them starts feeding on the chemicals of the nutrient medium. Then immediately the medium gets exhausted. Again you will be adding the nutrients, chemical. That is chemical medium is, that is the bacterial culture medium is again sterile bacterial medium is culture medium, sterile medium is again added without microorganisms, so that continuously without collecting them for our experimental purpose, you will be continuously loading the nutritive medium, so that the microorganisms will get the nutrients and they feed on the chemicals that are present in the culture medium, and their number increases, so that when compared to batch culture, in fed batch culture, the density, cell density increases. అంటే ఏంటి, ఈ మీడియంలో సెల్ నెంబర్ ఎక్కువ అయిపోతూ ఉంటుంది. బ్యాచ్ కల్చర్లో ఏం చేస్తున్నాం, 3 రోజులకు ఒకసారి షేకర్ ఆపి దానిలో తీసి 3 ఫ్లాస్క్లలోకి వేస్తున్నాను. కానీ ఇక్కడ ఏమీ చేయట్లేదు, సెల్స్ ఉన్నాయి, డివైడ్ అయ్యి ఒక 10 సెల్స్ వేస్తే 100 సెల్స్ అయ్యాయి ఫస్ట్ టైం. ఆ 100 సెల్స్కి మీడియం అయిపోయింది కాబట్టి మళ్ళీ మీడియం వేసాను, మళ్ళీ ఆ 10 సెల్స్ 100 సెల్స్ 1000 సెల్స్ అయినాయి, 1000 సెల్స్. ఈ 1000 సెల్స్కి మళ్ళీ మీడియం యాడ్ చేశాను, మీడియం ఎగ్జాస్ట్ అయిపోయింది. ఈ 1000 సెల్స్ 10,000 సెల్స్ అయ్యాయి, మీడియం ఎగ్జాస్ట్ అయింది. అగైన్ ఐ హ్యావ్ యాడెడ్ మీడియం. 10,000 సెల్స్ 1 lakh సెల్స్ అయ్యాయి, అగైన్ ఐ హ్యావ్ యాడెడ్ medium. That means in this 10 lakh cells are there. That means the cell density increases in the fed batch culture. Next point, in this fed batch culture the chemicals are added in such a manner that they should not become toxic to the cells, growth of the cells. Ante enti, when you are adding the nutrients you should be very careful that you have to sterilize it and add it. Once the medium in the culture vessel or fermenter is getting exhausted, ante adi ayipotunna time lo you have to add fresh medium, sterile fresh medium into the culture vessel, so that the cells will start eating on that and they will start increasing in size. Then they lead to lag phase continues to log phase by division and the number of cells increases. That means here the cell density increases compared to the batch culture. Second one is the nutrients are added in such a manner that they should not become toxic to the cells. This is about your fed batch culture. Okay, coming to the continuous culture. What is continuous culture? Continuous cultures means continuously log phase cells are maintained. Continuously log phase cells are maintained. This is known as continuously log phase cells are maintained, and this is known as continuous culture. Continuous cultures are of two types: closed continuous culture, open continuous culture. Closed continuous culture and open continuous culture, these are the two types of cultures. Coming to the closed continuous cultures, let us see. Suppose this is your container, this is your turbine inlet, you will be releasing the medium into this and you will be releasing the microorganisms. Now this is your outlet and this is your collector. Now when it is spinning, uniformly mixing of medium and microorganisms takes place, and these will help in your production of your product. It may be protein, enzyme, vitamin, whatever it may be, your lactic acid or glutamic acid or your penicillin, antibiotic penicillin or streptomycin, whatever it may be, they are synthesized by these cells. Once it is synthesized, the product is collected through the outlet. This is inlet. The product is collected through the outlet into a collector. Once the product is collected, it is filtered. You will get cells and filtrate. These cells are again added to this along with the medium. That means the cells are not used for our experimental purpose. Instead, the cells are filtered, and the cells that are collected on the filter, they are again added back into the bioreactor or fermenter or bioreactor. This is known as closed continuous culture. This is known as closed continuous culture. Coming to the open continuous culture, in this the medium is added and the microorganisms are added, and the microorganisms and medium and microorganisms are uniformly mixed. That is the turbines mix uniform, that is the turbines uniformly mix the medium and the nutrients, so that these cells enter into lag phase and log phase and they start producing the product. Once the product is produced, it is collected into the collector. Once it is collected, this collected filtrate is used for our experiment. It is not again added into this. Instead, it is used for our experimental purpose. This is known as open continuous culture system. Okay. Closed continuous culture system means the collected cells are again added, resuspended or re-added into the fermenter for continuous growth. This is known as closed continuous culture system. And when the cells are, after collecting, the cells are used for our experimental purpose, that is known as open continuous culture system. Okay. Now we are interested in open continuous culture systems. The open continuous culture system is See here, the open continuous culture system is of two types, chemostat and turbidostat. Already we have discussed this topic in your third semester. Now what is chemostat? I think you won't remember, you won't be remembering. Now what is chemostat? Chemostat and turbidostat. Chemostat is a open continuous culture system, or chemostat is a open continuous culture system. This helps in maintenance of log phase cells continuously. It is known as open continuous culture system, that is chemostat. In this chemostat, see here, this is your reservoir, and this is connected to your culture vessel, and this is connected to your collector. This is your culture vessel, or you can call it as fermenter. Okay, this can be called as a culture vessel or fermenter or bioreactor. Now this consists of a medium, and the medium is released into this, and microorganisms are released directly into this, so that the microorganisms are released like this. Now see here, once it is released, the reservoir culture medium, sterile culture medium is released into this. In the sterile culture medium, we will be adding all the media chemicals. We will be adding all the media chemicals in excess amount. Suppose magnesium sulfate is there. Its requirement is 10 grams, but I am adding 15 grams. Sodium molybdate 0.5 grams is required, but I am adding 2 grams. Like that, all the chemicals are added in excess amount, so that the media chemicals should not deplete or exhaust during the course of fermentation. During the process of fermentation, the media chemicals, they should not undergo depletion, or they should not get depleted or exhausted because of the feeding of the microorganisms on the chemical medium. The excess chemicals are added, excess amount of all the chemicals are added, excluding one special chemical which is very much essential for the cells to enter into log phase or division phase. So you will be adding all the chemicals except calcium chloride. Calcium chloride is very much important for the growth of the cells, that is division of the cells, so that cell division chemical, that is calcium chloride, is not added into this reservoir chemical medium, or if it is used also, only 4 grams is required, but instead of 4 grams you are using 0.0004 mg. అంటే ఏంటి, వేసామా లేదా అన్నంత కొంచెం వేస్తున్నాము, లేకపోతే అసలే వేయట్లేదు. దిస్ కాల్షియం క్లోరైడ్ ఈస్ నాట్ ఎట్ ఆల్ యూజ్డ్ ఇన్ ది కల్చర్ మీడియం, అండ్ దిస్ ఈజ్ హైలీ ఎసెన్షియల్ ఫర్ ది సెల్స్ టు ఎంటర్ ఇంటూ లాగ్ phase. Now all the cells, these are the cells, see here, I will draw here, these are the cells which are released, small cells, these are released into this medium initially. Now these cells, they feed on the nutrients which are excessive in amounts in the culture vessel or fermenter or bioreactor. Now they increase in size, see here, all the cells increase in size, like this they increase in size. Once they increase in size like this, that means the cell size increases, because all the cells, they are getting good nutrition, so they feed on the nutrients. Though they are feeding on the nutrients, the nutrients in the medium, they are not getting exhausted, because we have added excess amount of all the chemicals. So the cells start feeding on the nutrients or the chemicals of the culture medium, and their cellular content increases, their mitochondria, endoplasmic reticulum, Golgi complex, vacuoles, whatever it may be, all its numbers increases, and the cell size also increases. When the cell size has increased, this is known as a lag phase, but they cannot enter into log phase, which is useful for our continuous culture system. We require log phase cells. So we require log phase cells. For that reason, see here, these cells, they have not entered into log phase, they are still in the lag phase only, because that special chemical is not there. Once all the cells enter into lag phase, now I am adding this calcium chloride into this culture vessel directly. When it is added into the culture vessel, immediately all the cells start grabbing on that, because they are starving for that chemical. అంటే ఏంటి, అవన్నీ సైజు పెద్దవైపోయి ఎదురు చూస్తున్నాయి, నేను ఎప్పుడు డివిజన్ ఫేజ్లోకి వెళ్ళాలి అంటే నాకు కాల్షియం క్లోరైడ్ కావాలి. ఆ కాల్షియం క్లోరైడ్ లేదని అవి ఆకలితో ఉన్నాయి, అంటే దే ఆర్ స్టార్వింగ్ ఫర్ దట్ కాల్షియం క్లోరైడ్. And once a calcium chloride is added into that culture vessel or fermenter or bioreactor, immediately all the cells will come near to that, onto the surface, and they start grabbing that chemical hastily. అంటే ఏంటి అంటే, మీరు ఒక ఫిష్ పాండ్కి వెళ్ళి కొన్ని మరమరాలు తీసుకెళ్ళి ఇట్లా వెయ్యగానే పాండ్లో ఉన్న ఫిష్ అన్నీ ఒక్క చోటకి ఇట్లా దగ్గరికి వచ్చేస్తాయి ఆ మరమరాలు తినడానికి. అట్లనే ఆ కెమికల్ వేయగానే అన్ని సెల్స్ దానికోసం ఆకలితో ఉన్నాయి కాబట్టి, ఆ కెమికల్ గురించి ఆకలితో ఉన్నాయి కాబట్టి, స్టార్ చేస్తున్నాయి కాబట్టి, ఆ స్టార్వింగ్ సెల్స్ అన్నీ పైకి సర్ఫేస్ మీదకి వచ్చి ఆ కెమికల్ని గ్రాబ్ చేసుకొని తినేస్తాయి. వన్స్ దే హ్యావ్ దట్ కెమికల్, ఇమీడియట్లీ సైమల్టేనియస్లీ ఆల్ ది సెల్స్ విల్ ఎంటర్ ఇంటూ లాగ్ ఫేజ్. See here, simultaneously all the cells will enter into log phase. That is when the cells enter into log phase simultaneously, immediately after adding this calcium chloride, within half an hour or 1 hour, based on the time of the standard time of this reaction, this knob, which is an automatic opening, this automatic opening will open and all the cells are collected here, consisting of small cells. All the cells are now in log phase, and the log phase cells are collected here, and the collected log phase, all are log phase cells, and these log phase cells are used for our experimental purpose. This is known as your chemostat. So that means chemostat is a open continuous culture system that maintains continuously the log phase cells. In this culture vessel, one of the chemical, all the chemicals in the culture medium, all the chemicals of the culture medium are added in excess amounts. All the chemicals are added in excess amounts in the culture medium, except one chemical which is either not added completely, or which is added, if it is added also, it is added in very limited amounts, so that all the cells will enter into lag phase. Their cell size increases, cellular content increases, but they are not ready to enter into log phase, as that chemical is missing in that culture medium. Once that chemical is added into that culture medium, immediately all the cells, as they are starving for that chemical, all the cells, they start feeding on that chemical, and simultaneously, that means that is at the same time all the cells enter into log phase. Once they enter into log phase, these cells are collected by the automatic opening that opens, that knob gets opened, and all the cells are collected, and these cells, they are used for our experiment. This is known as your chemostat. Now coming to the turbidostat. Turbidostat means, see here, turbidostat is also having the same setup. See here, in this turbidostat, this is your culture vessel. See here, this is your culture vessel. Now see here, in this side lamp is present. This side lamp is present. On other side, it consists of a photocell. On other side, it consists of a photocell. What is it? See here, on other side it consists of a photocell. Now the photocell is a equipment that senses the light. It is known as a photocell. That means that senses light. So here medium is there, turbine is there. Now small cells are released. 10 to 20 cells are released into the culture medium. Reservoir chemicals are released. In this, all the chemicals are added. Not only the one chemical is not added or limited, it is not at all limited. All the chemicals are added. We are not stopping any chemical, we are adding all the chemicals, and these chemicals are released into the culture vessel or your fermenter or bioreactor. And now what happens, the lamp is switched on. Once the lamp is switched on, the light will pass through, see here, the light will pass through it, and it will reach the photocell, and the photocell will sense the light, and the pointer which is here, it will move like this. The pointer which is here initially at 0, it will move to this like this. That means as the medium is diluted medium, that is the medium consists of single cells, small cells, and their cell number is less and the cell size is also small. So the light without any prevention, it will pass through the culture medium. As it is a glass vessel, it reaches the photocell, and the photocell sends the light. Once the cells, they start feeding on the nutrients that are present in the culture medium. Now the cell size increases. See here, the cell size increases. Once the cell size increases, they will enter into log phase. Once they enter into log phase, the number of cells increases in the culture medium, and the medium becomes very thick and concentrated. See here, the medium becomes like this. Once the medium becomes like this, immediately for these cells, see here, for these cells, it will be, the light cannot pass through this culture medium because the medium has become concentrated solution. It will not allow the light to pass through it easily. The light cannot pass through it easily. Second one is, if light is passing through the culture medium, one or the other cells will stop the light ray. It comes in the way of the light ray and it will stop the movement of the light ray, so that the photocell cannot sense the light. So once it is not sensing the light, again it will come to 0. That is, the pointer will fall down and it will reach 0, or at least 1 or 2 or 0, it will reach 0 or 1 or 2, so that the photocell is not sensing the light. Once the photocell is not sensing the light, it is an indication that this knob gets opened and all the cells are in log phase. That means once this photocell is not receiving the light, or once the photocell is not sensing the light, it is an indication that the culture vessel or the fermenter or bioreactor consists of all log phase cells. Continuously log phase cells are there, and these log phase cells can be collected into the collector. This is known as a turbidostat. Turbidity means thickness or concentration. Turbidostat means turbidity. అంటే చూడండి అమ్మ, మామూలు ఒక గ్లాస్ వాటర్ 2 గ్లాసులు తీసుకున్నాను, ఒక గ్లాసులో మంచినీళ్ళు తీసుకున్నా, ఇంకో గ్లాసులో ఏమో నీళ్లు తీసుకొని దానిలో ఇంత మట్టి కలిపాను. అప్పుడు ఏమైంది, ఈ వాటర్ ఏమో ట్రాన్స్పరెంట్ వాటర్, ఇటు నుంచి అటు గ్లాసులోంచి చూడగలుగుతున్నాం. ఈ వాటర్ థిక్ అయిపోయి, టర్బిడ్ అంటే మురికి నీళ్ళలాగా, మట్టి నీళ్ళలాగా అయిపోయి ఇటు నుంచి అటు చూడడానికి ఏం కనపడట్లేదు. సో సీ హియర్, so turbidostat is a open continuous culture system consisting of continuously log phase cells, and this is working on based on the turbidity of the culture medium. Once the medium has become turbid or very thick or concentrated, that is an indication that the culture vessel or the fermenter or bioreactor consists of all log phase cells, and the automatic opening will open, this automatic opener will open, and immediately the cells are all the log phase cells are collected and they are purified and used for our experimental purpose. This is known as a turbidostat. This is another type of fermentation, that is your continuous open type of continuous fermentation system. Now coming to the next one, that is aerobic fermentation and anaerobic fermentation. దీని గురించి ఏం పెద్ద చెప్పేది లేదు. Aerobic fermentation means in this oxygen is used. In aerobic fermentation, in this oxygen is used, and these oxygens, when it will be passing through the sparger into the culture vessel, and it will be helping in mixing. These aerobic fermenters, they may be mechanical or non-mechanical, and they consist of turbines or impellers or blades or stirrers. If not, they are just like airlift fermenters. The air will mix the medium and the air mixes the medium and the culture. The air uniformly mixes the medium and the microorganisms. This is where oxygen is used in the culture medium. This is known as aerobic fermentation. It is useful in the production of monoclonal antibodies, vaccines, etc. Anaerobic fermentation, in this carbon dioxide is used through the air pump, and here oxygen is not useful. And this anaerobic fermentation, the microorganism itself releases the carbon dioxide, and that carbon dioxide is used for its survival, and division and increasing in the number of cells, that is log phase. So that is known as anaerobic fermentation. Examples of these microorganisms are Clostridium, methanogenic bacteria, this helps in the production of biogas. Beer, wine, this is anaerobic fermentation. The production of biogas, beer and wine, this is your anaerobic fermentation. That means types of fermenters are solid state fermenters, submerged fermenters. Submerged fermenters are of three types: batch culture, or solid state fermenter is also known as shallow fermenters. They are useful in the growth of your mycelial microorganisms. Submerged fermenters are of three types: batch culture, fed batch culture, continuous culture. Continuous culture is of two types: closed continuous culture system, open continuous culture system. Open continuous culture system is of two types: chemostat and turbidostat. Chemostat and turbidostat already we have discussed. Now coming to the aerobic and anaerobic types. Aerobic fermentation is a type of fermentation. In this, oxygen, in the presence of oxygen, fermentation takes place, and anaerobic fermentation in the presence of carbon dioxide, that is in the absence of oxygen, your fermentation takes place. This aerobic fermentation example is monoclonal antibody production, vaccine production. ఇప్పుడు మన కోవిషీల్డ్, కోవాక్సిన్ అవన్నీ ప్రొడక్షన్ aerobic fermentation. Anaerobic fermentation is biogas production, beer production, wine production. All these are anaerobic fermentations. Is it okay, all of you? So today we'll stop here in these types of fermenters, and tomorrow we'll continue with the next topic. That is, what are the media components that are used in your culture medium, and how to satisfy our cells with how to banjayman. So tomorrow we'll see the next topic, that is your types of formulation of the medium. So we'll stop here today and we'll continue tomorrow.